Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 56, and it's going out live on April the 24th. Lots to get through this week, so no chat, which is probably just a relief to all the listeners. Last week we had a conversation with Jason Bock, CEO and co-founder of CTMC, and with Eledon President and CSO Steve Perrin. This week our conversations are with Bruce Leuchter, co-founder, president and CEO of Neurvati Neurosciences and GRIN Therapeutics, and with Daniel Vitt, CEO of Immunic. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, sponsoring a podcast episode, or for event coverage. First, it's time to go through some of the headlines of the articles you may not have read this week over at deeptechdigest.com. An AI-powered biochip can detect genetic markers in 20 minutes, and I can't even find my permanent markers after an hour. A CRISPR variant selectively targets tumor DNA, and researchers have developed an inhaled therapy to reduce lung damage from severe infections. A new technique maps cancer drug uptake inside living cells. There's a new stem cell therapy for type 1 diabetes, and Resolve Therapeutics and Institut Pasteur are collaborating to identify cell-free RNA triggers in autoimmunity. My Tomorrows and Rare Disease Research are partnering to improve access to rare disease clinical trials. A mechanism has been discovered that opens a new era for targeted cancer therapy, and UCB has added an epilepsy drug through the acquisition of Neurona Therapeutics. A newly launched company is planning to replace daily injections with an implant. There are positive first-in-human data for a treatment for IVF-related embryo implantation failure, and you can read all of these and plenty more at deeptechdigest.com. Alright, let's keep things moving, and first off this week, Immunic is a late-stage biotechnology company pioneering the development of novel oral therapies for neurologic diseases. The company's lead development program, vidofludimus calcium, is currently in phase 3 clinical trials for the treatment of relapsing multiple sclerosis, with top-line data expected by the end of 2026. To tell us more about the program and the company is its CEO, Daniel Vitt. Good morning, Daniel. Thanks a lot for being a guest on this week's podcast. And maybe to start, you could tell us a little bit about the company.
At Immunic, we are developing new oral treatment, safer oral treatments for treating chronic inflammatory and autoimmune diseases, and specifically in the space of neuroinflammation and multiple sclerosis, where we have our phase 3 asset ongoing there, generally with the idea to focus on the key unmet medical need in those indications.
And could you tell me a bit more about the conditions that you're addressing?
Yeah, maybe the most important is, as I said before, multiple sclerosis. In multiple sclerosis, we have our lead asset, vidofludimus calcium, which is designed to really target the key unmet medical need in patients suffering from MS. And this includes two big groups of patients in relapsing and in progressive MS. And I think the key strength of vidofludimus calcium and the key differentiator is that on the one hand, it is a very easy to use oral once daily tablet with an outstanding safety and tolerability profile, so easy to use for the patient and also for the prescribing neurologist. But also is addressing for the first time a direct neuroprotective mode of action by activation of a target called Nurr1. And that, I think, makes it different from what is established in treating multiple sclerosis today.
Could we maybe run through some of the programs that you have, starting with 838?
Yeah, let me start with IMU-838, which is, as the INN code, vidofludimus calcium. So vidofludimus is in clinical trials and different indications ongoing. One is we are running a phase 3 clinical study in patients with relapsing forms of multiple sclerosis, RMS. And these are two identical twin studies going on globally, and we are expecting to read out the data from these two phase 3 studies at the end of this year, so pretty soon. And this is then planned to submit an NDA mid of next year, and then if things go well, to launch vidofludimus calcium in RMS mid of 28. In parallel, and as I said before, given this unique mode of action and being able to directly target neuroprotection, we also have performed a phase 2 clinical study with IMU-838 or vidofludimus calcium in patients with progressive MS. And we have seen quite amazing clinical data there that on a numerical level, we have seen a reduction of confirmed disability worsening, which is the unmet medical need, basically in all forms of MS. We have seen a reduction in the total study of more than 24%. And specifically in PPMS, in primary progressive MS, we have seen 31% reduction of disability progression, which is outstanding, which is really has the potential to change the way how we treat the disease. And based on that, we decided to go into a phase 3 clinical study in primary progressive MS as soon as possible. So the preparation of starting that phase 3 study are currently ongoing. This is IMU-838, and I told you it also has shown good effects in GI disorders like ulcerative colitis, but here clearly the focus is on bringing that to the finish line in MS.
And what does that mean for patients with MS? Does it reduce their symptoms? Is it a way of stopping the disease? I mean, what are you looking at for the end results for patients here?
This is the most important question for us here, but also for the patients. So I think the biggest unmet medical need in multiple sclerosis is really progression of disability. And then, as I said before, there are these two big things you observe in MS patients. One is relapses. It's an episode, it's a limited episode where patients get worse and then they improve in the remission phase again. So this is an on-off thing. It is typically more in the early stage of disease and more in this relapsing form of MS. The second thing, and this is something which has more or less emerged in the last couple of years, there's a little bit of an independent process ongoing we call PIRA, so progression, which is independent of relapse activity. And this PIRA, in fact, is really linked to just progression. And this is something we observe in all forms of MS, and this is what patients are worried about. So with our mode of action as a Nurr1 activator and that direct neuroprotective effect, we think that Vido has the unique potential to slow down or even stop progression. And an interesting piece here of data is that in our phase 2 study in progressive MS patients, we have even seen improvement. So a statistically significant post-hoc analysis here showed that the likelihood of patients on treatment in the Calibre phase 2 study was 2.4 times higher to improve compared to the placebo group. That is something interesting and something new, and I think it's something which also excites patients and doctors because it could ultimately lead to the situation where a drug like vidofludimus calcium can really not just stop or slow down disease progression, but also improve the physical ability of patients.
Does that mean that the timing of when you were first able to administer the drug makes a difference? If you can get to somebody earlier along that MS pathway than later?
I think that's definitely a point. And I think all studies have shown that the earlier you start, the better for the patient because you want to stop destruction. In this case, I think it really depends a little bit on the specific patient situation, prior treatments, disease status of the patient, so baseline EDSS score, so the disease activity or disability score of the patient. And typically, I would say it's good to have a safer drug because it allows you to use it earlier on a risk-benefit assessment by the doctor, right? The treating physician is important in the discussion with the patient. What can I do? What options do I have? And what's the balance between effect and side effect? And a drug like IMU-838 could really change that, the optionality here. Having an oral, easy-to-use drug, which is pretty safe and well tolerated, lowers the hurdle of the patient and the doctor. But secondly, also allows patient to adapt the treatment, which is maybe specifically beneficial on preventing disability progression earlier than you would anticipate in the current treatment landscape.
Do you see this as being something that's a standalone treatment or utilized in combination with other drugs?
I think initially as a standalone treatment, but this is more linked to the current treatment situation because the current available treatments are mainly, or most of those are linked to increased risk of infections. And that includes activation of John Cunningham virus infections, which may lead to PML. And PML has around about 50% likelihood of death of patients. So you don't want to increase that risk. That's the reason why neurologists typically don't combine current immunosuppressive treatments. Vidofludimus calcium is a little bit different. So with its, on the one hand, Neur1 activation and as a second mode of action, DHODH inhibition, which is anti-inflammatory, it may be the perfect molecule to be combined with something else. And I could envision in the future that you would maybe try it together with, for example, an anti-CD20 therapy.
And I guess other companies are constantly working on new treatments as well. So hopefully other new drugs are developed at the same time. So,
Yes, specifically if it comes to neuroprotection, I think everything which is developed in the future also targeting direct neuroprotection may be a good combination partner as well.
All right, so maybe moving on, you could tell me a bit more about IMU-856.
The IMU-856 is a quite unique molecule. It is targeting a protein called sirtuin-6. And in the phase 1 clinical study, we also tested it not only on healthy volunteers, but also in patients suffering from celiac disease. So that the drug is targeting the barrier and it is able to restore a proper functioning intestinal barrier by restoring the natural renewal of the epithelial layer in the gut. And we have shown in the phase 1 study that it is able to protect the destruction of villus structure in patients with celiac disease. Second, we have also shown that the drug is able to restore or improve the uptake of nutrients like vitamin B12, for example. So a functional restoring. And we believe that could ultimately really change the way we treat celiac disease and other GI disorders. However, given that we, based on the recent big financing we have done, really focus right now on completion of this phase 3 studies in MS, it had a lower priority, and we are more or less strategically more focusing on the neurology space right now. So so next phase 2 studies are subject to further funding there and maybe even partnering on that asset as a next step forward, and then to ultimately go into phase 2 studies in celiac disease or other GI indications. Which brings me maybe to the third molecule, IMU-381 and other things. I think what we are currently doing in our discovery research to develop the platform we have developed over the last years on the neuroinflammatory pathways, and specifically Neur1, as we own that target more or less right now. We have a lot of molecules which have been investigated in the context of Neur1 activation systemically and in a central fashion. So we are exploring different ways to leverage that platform with 381 and other molecules and to go further here in neurodegenerative diseases, including MS, but also Parkinson's disease and others. It's a huge set of indications where you could leverage such a platform. And I believe that Neur1 is a very interesting, cool target where we will hear a lot of things, not just from us, but also from other companies in the future. But right now we own that space and we want to play that card right now, having a good IP position, several chemical series of new molecules with different selectivity profiles. That makes it quite interesting.
You mentioned 381. What's that one?
Yeah, that's a molecule which is a Neur1 activator, and we are exploring here the way towards clinical development as quickly as possible right now.
So with the platform that you mentioned, is that a priority, is to come up with as many potential drugs as possible and then partner on those?
I think yes, Immunic has brought vidofludimus calcium now to phase 3 with a new target, Nurv1, which is, I think, interesting per se for all these different neurodegenerative diseases. And this is a huge set of indications where you can leverage that. And it is a value story at the end because we own that chemistry and the IP. It is important to make that visible and to get forward with these things. And from a strategic point of view, it's important to get as quickly as possible molecules with different profiles into clinical stage from that platform. And of course, that's an opportunity to partner in different indications, but also to continue development in maybe one or two other molecules in the neurospace on our own. So it gives us optionality, which I think is a key value driver here for that.
And how difficult is that from, I don't say from an ethical point of view, but how difficult is it when ultimately you want to help patients and you have many different indications and you're developing others? How do you kind of push all of that forward so that patients are being served at the end of the day?
Every decision in pharma biotech development is an ethical decision, and luckily the medical need usually is the ethical driver as well. So unmet medical need is key, and I think neurodegeneration is one of the key things these days. It's a huge unmet medical need everywhere, from Alzheimer's, dementia, Huntington's, Parkinson's disease. So all of these things need proper new treatments, and there is little available right now. Learning more about Nurv1 activation, and we are very heavily working on this in our research teams. We think that this is one of the interesting, outstanding new targets you can leverage on all of these different indications. And then at the end is the question, what's the smartest way to bring a molecule forward to help these patients, right? So, and this is part of the process we have ongoing.
Why do you think it is that a lot of companies focus on symptom control rather than actually addressing the disease and disease modification?
Lack of ideas, I think, how to do that. So it's more a biology point. There are some interesting findings and some interesting new concepts around, but you need to find a molecule which is really doing the job. And for example, for Nurv1, it's known for 10 years maybe that this is one of the cool regulators or master switches for neurons and cell fate of neurons. But it took quite a time to find a molecule which is activating it. It is easy to find an inhibitor of something like that, but difficult to find an activator. So it's sometimes a scientific problem to find the right molecule. And that's maybe true also for other approaches. And sometimes you find a molecule, you don't know how it is working. There's a lot we need to do as scientists to understand the connection between a molecule, its mode of action, and the clinical consequences of using that.
And how are small molecule drugs being re-engineered to address some of these complex immune diseases?
It's still medicinal chemistry. You get support of a lot of tools, but at the end, it's synthesizing the right molecule. And then have the right to ask the right questions in the lab. What are the key parameters you want to ensure that the molecule has? In our case, it's really vidofludimus calcium is a dual-acting molecule. It's acting on Nurv1 and then DHODH. And one step, for example, for a 381 would be to make sure we have a molecule which is selective just for Nurv1, so that makes it maybe an interesting treatment for Parkinson's disease, for example. But also you can look on molecules which are more selective towards DHODH or which are more systemically available, where you, for example, want to target immune tolerance as a different concept. So this is more engineering by modifying based on the structural activity relationship you identify in the lab. And this is a knowledge business. So we are synthesizing molecules and we're testing them as broadly as possible, and we're generating data, and we're then leveraging that data on a broad scale to decide what is the next modification the team is doing to the molecule to bring it forward.
Has artificial intelligence helped with that process in terms of the ability to find molecules and also to compact timelines?
Honestly, I think vidofludimus calcium was maybe one of the first molecules coming from AI in the past, because it was at that time was called in silico screening, and we used a technology which was based on support vector machine learning algorithms. So yes, it does. But in chemistry, I think for optimizing molecules, it today plays maybe not that role, but it plays a big role in the biology. I think the team is really leveraging AI a lot in more or less in analyzing the pharmacological landscape and the targeted biomarkers and the signals we can detect, and to bring it from preclinical to clinical stage. So it's more data driven at that point, but in a design point, the predictive power of computer tools, including AI today on single modifications, is limited, I think. And because it has so many different aspects, it's still perceived to be a more straight classical medicine and chemistry approach in the optimization.
And so with all of the work that you're doing, what do you see as the next, short term and a bit more medium term for the company?
Clearly bringing vidofludimus to the market. That's our mission right now. We want to see it on the market, and we want to not only see it on the market in relapsing MS, as planned in 28, but also in primary progressive MS. I think patients deserve to have a treatment for treating their progression and stopping progression. This is my personal vision for the company, to help these patients. And then as a second step, to leverage the platform to also help patients with other indications. So not just limit it to MS, but broaden it and build on that. So this is, I think, what we are working on. We are committed on that. We raised 200 million plus another 200 million option just recently this year to be able to do that. We are building a very good team here around the planned launch of the drug and making also the neurology world more familiar with what we're doing here. A new mode of action always needs education and to really make sure people get what's the difference here, what's new about the molecule. And so this excitement is huge here among the company, investors, and the community here. And also, I think if you talk now these days with treating physicians, there is excitement about something new coming. Also considering that in the MS space, the BTK inhibitors had a little of a hard time and did not deliver what was promised before. So I think that is another reason why there's excitement about what we are doing.
And does that excitement extend to, clearly you must go to a lot of events? Are you seeing that from the events you go to?
Yes, absolutely. We go on every neurology event which has to do with MS right now. The team is there. We are building a pretty good team really to talk to the investigators and talk to the treating physicians. That's key. It will be, I think, driving force will be a launch in the United States first, and then followed by Europe and other jurisdictions. But as a small biotech, you don't have the established workforce as a big pharma has to do everything in parallel. But it's good to have the United States as a starting market for that drug and then to expand from there.
And I assume that you'll be doing lots of studies as you go once it's launched to track it.
Yes, and you mentioned some of the key questions. Can it be combined? Is it safe to switch from another treatment here? So there are a lot of things we want to address, and also looking on biomarkers. What does it mean? Can we measure neuroprotection with an easy biomarker? There's something which is new and maybe something which is more scientific interest, but it may help to design the next treatments and also maybe to find the right patients in the future which are specifically well responding to the treatment. So there's a lot we want to do. The most important message, I think, is here. We spent the last years on bringing this molecule from phase 1 into phase 3. And now I think it's a big step for the company and a transformative step for the company to move from an R&D organization to an organization which is able to successfully commercially launch a drug in that big indication and actually to change patients' life. And knowing so many MS patients, I think it's the good thing about what we are doing here is to help these patients to continue with a normal life as long as possible. This is our mission, and this is what I think we as a company want to achieve.
Super news for the MS field, although as with everything else, solutions and treatments can't come soon enough. We're switching now to learn about GRIN and GRIN-related neurodevelopmental disorder with the co-founder, president and CEO of Neurvati Neurosciences and GRIN Therapeutics, and that is Bruce Leuchter. Hi Bruce, great to have you with us to learn more about GRIN. So maybe to kick things off, you could give us a bit of background on the company.
Sure. We have an interesting construct. So we are a Blackstone-backed organization. All the work we do is funded by Blackstone Life Sciences, who has a deep and longstanding commitment to neuroscience. And there are folks on the Blackstone Life Sciences team who've trained in neurology, who've started companies in neuroscience. And so this was a logical extension of that baked interest. And Neurvati was created to serve as an operating vehicle. So when you think about Neurvati, think about it as the people. What is the platform technology? And I say the platform technology is the people, because the idea behind forming Neurvati was to create best-in-class expertise in an organization that can prosecute mid to late-stage therapeutics across the neuroscience landscape. The idea being that when we identify a clinical stage molecule with data, likely sitting at a mid-cap or large-cap pharma who's engaging in portfolio reoptimization or has a desire to externalize a program, we can license that in or acquire the asset and form an SPV or affiliate co around that molecule in that program. And that affiliate co sits separate to Neurvati, so sits alongside Neurvati. And the people at Neurvati Neurosciences are deployed against the affiliate co to develop the asset in mid and late stage studies. And the first example of that is GRIN Therapeutics, which was formed in September 21 at the same time Neurvati Neurosciences was formed. So the two companies were formed simultaneously for obvious reasons, because the people that do the work in the affiliate co sit at Neurvati. So we identified a molecule that was within UCB Pharma. It was a molecule that had data, pretty substantial amounts of data, which gave us confidence in the safety, tolerability, dosing protocol, PK of the molecule, and had some data in a rare pediatric epilepsy called infantile spasm. So we looked at those data and we said, clearly, this molecule is active. And then we stepped back and said, where can it be deployed to realize the maximum clinical benefit? And we came to GRIN-related neurodevelopmental disorder for that reason, because the molecule itself, radiprodil, is a negative allosteric modulator at the NR2B subunit of the NMDA receptor. And these GRIN children and adults have a mutation on the GRIN gene. The GRIN gene encodes for the NMDA receptor. So when they have a gain of function mutation, they have hyperactive NMDA receptors, and we go in with radiprodil and downregulate that activity. And that should have impact. Our thesis from the get-go was that that should have impact on the broader disease. Of course, seizures are an easier thing to measure in an open label study with a small number of patients. And we did that in our phase 1b/2a study, but we were also quite keen on figuring out how effective it could be across the broader disease. So we had a cohort of patients whose seizures were well controlled, and we saw improvement. The longer they were on drug, the more improvement we saw across non-seizure endpoints. And that's what got us to the level of conviction that we had as we were approaching regulators to organize around our phase 3 study, which we just kicked off a couple of months ago, global phase 3 in the US, Canada, Europe, and the UK, and several countries in Asia Pac. So it's been a whirlwind experience over the course of the last 4.5 years or so, and we're really excited about where we've gotten the program to. What's been most gratifying, as you might imagine, is the reaction from the community. And this is a community that is relatively new compared to other patient advocacy groups who've been around for decades. This is a group that was formed relatively recently. The disease itself was only named in 2014. So you can appreciate that the efforts from the parents and the caregivers and the patient advocates is relatively new, but they've done an amazing job organizing themselves and collaborating with us to get us to the point we're at. And they've been thrilled with the engagement from industry just at the highest level, understanding that folks who develop drugs and investors who finance those development activities are focused on their disease. And then the impact that we saw in the phase 1b/2a study was really terrific and has catalyzed the community really across the globe, hence my upcoming trip to the UK for the GRIN UK conference in May.
To take a step back a little, could you go into a bit more detail on GRIN-related disorders and the differences between the different types?
Sure. So GRIN-related neurodevelopmental disorder is a group of disorders that present the same way in essence, but are driven by different mutations on different GRIN genes that encode for different subunits of the NMDA receptor. So you have GRIN2A, GRIN2B, GRIN1, and GRIN2D. Those are each
different genotypes of GRIN-related neurodevelopmental disorder. And they present very similarly. It's a constellation of symptoms where they have neurodevelopmental delay, they've got behavioral dysregulation, sleep-wake cycle dysfunction. Many require 24-hour supportive care. Some are wheelchair-bound, some have feeding tubes, many are nonverbal. It's a constellation of classical neurodevelopmental symptoms that you might see in other neurodevelopmental diseases or other developmental epileptic encephalopathies. And they present pretty similarly. So take a 2-year-old child who has had neurodevelopmental delay and behavioral dysregulation plus or minus seizures. They present to the pediatrician. The pediatrician suspects that there's a neurodevelopmental disease, and that neurodevelopmental disease needs to be evaluated by a specialist. So these kids end up seeing pediatric epileptologists, or they see pediatricians specializing in neurodevelopmental disease, or geneticists. And when they suspect that there's a neurodevelopmental disease, because of where we are today in drug development, where you see a number of different disease-specific therapies coming through late-stage development, these kids need a genetic diagnosis in order to determine what medicine is best for their underlying disease. So they submit to whole exome sequencing. And a caveat on that for a moment is that whole exome sequencing in prior years has been a difficult thing to access for many patients around the globe, certainly in the US. That's changed quite a bit from the standpoint of technological advances that have enabled higher throughput, lower cost for whole exome sequencing. And the commercial efforts that have been undertaken in that regard have yielded a lot more information for patients and families, but also for physicians and certainly for drug developers as we try to zero in on the underlying cause of the disease. So they do that. These kids will get a whole exome sequence. When they get it, it'll be clear that they have GRIN-related neurodevelopmental disorder because they'll see a mutation on the GRIN gene, similar to what they might see if patients had CDKL5, PCDH19, SCN8A, SCN2A, SCN1A. These are all genetically defined neurodevelopmental disorders and or developmental epileptic encephalopathies. But when they do have a GRIN mutation, they now are eligible for us to get involved and determine and characterize what that mutation is. Is it a gain of function mutation? Is it a loss of function mutation? Is it indeterminate? And if these patients have a gain of function mutation, they become eligible for our study. And we aim to enroll them in our ongoing phase 3. So that's the patient journey. And the good news is that these patients do present because the conditions they suffer from are highly morbid. The good news is also now that they can get a definitive diagnosis. I think the frustration from many of these families and caregivers historically has been that they go from doctor to doctor attempting to land on a specific diagnosis and struggle to do so, in part because of visibility for certain diseases like GRIN prior to 2019. And now they have the opportunity to come and get whole exome sequencing, which has done a great service to the broader space, really all constituencies, families, caregivers, drug developers, clinicians, et cetera. So we've been privileged to be a part of that. And it does take a village. So when you go after a disease that heretofore has not had any approved FDA treatments, and the only treatments have been empirical, patient comes in with seizure, give them an anti-epileptic. They come in with behavioral dysregulation, use a psychoactive medication. But they haven't had an opportunity to get a disease-specific therapy, and that's what our mission is, that's what our goal is. And as a broader commentary, I think that's where the space is going. The ability to make these definitive diagnoses by way of genetic testing gives us an opportunity to do more than just provide symptomatic treatment empirically. And our goal is to be at the tip of the spear of that effort. And what we can see from our peers in the space, people believe in that approach, and physicians believe in it too, and the level of excitement is pretty high.
What are the numbers like for the number of people with these conditions, either globally or in the US?
So GRIN-related neurodevelopmental disorder is driven by a de novo point mutation. So it's got, in essence, equivalent epidemiology across the globe. That's the expectation. And to the extent that it's been studied to this point, it proves itself out. So there have been a number of studies driven by academicians, clinician scientists who focused on this disease for some time. And those epidemiological data support the fact that what you see in different geographies is just about the same epidemiologically, given that it's a de novo point mutation. And when we look at real world data, we determine that the population of patients with GRIN in the US is about 30,000 patients. That includes gain of function, loss of function, and indeterminate patients. and it includes pediatric patients and adult patients. Our job is to work on that funnel to understand the population of GRIN-related NDD with gain-of-function mutations. And when we look at real-world data in that regard, we see about 15,000, so half the patients experience a gain-of-function mutation. What do I mean when I say real-world data? So one of the exercises we undertook was to go to GeneDx, who is the leading whole exome sequence provider, public company that does these studies, whose throughput has gone up substantially over the course of the last few years, given the ability to access the technology, in part through industry sponsorship, in part through a willingness to pay by government payers and private payers. And we asked them for their hit rate of GRIN mutations. We said, if you, over the last 60, 70,000 studies that you've done, what are the number of hits for GRIN mutations in your patient cohort? We did a similar thing around Dravet. We said, well, in order for us to make sense of that hit rate, can you please look at a similar hit rate analysis for Dravet? And what we saw was that the hit rate for all of GRIN exceeded the hit rate for Dravet, which of course is driven by an SCN1A mutation. So we've taken half of that and have gotten to this number of 15,000. When we look at peer-reviewed publications that have been driven by the experts in the field over the years, those numbers triangulate. So about 12 to 15,000 patients is the expectation in the US. When we've been able to drive awareness amongst the patient advocacy communities and physicians and clinicians who treat these patients, we see more patients coming into the fold. And then we see the efforts that exist away from us, driven by the general gravitational force toward whole exome sequencing, and we see hit rates increase. So I think ordinarily in rare disease, the expectation is that as viable therapies come online, visibility around diseases go up, diagnosis rates go up, and more patients get treated. When you couple that general phenomenon with the rising tide in whole exome sequence, we expect to see a compounding effect. We're bullish on the ability to reach more patients, and it's always a challenge when you are targeting a disease that hasn't had clinical development against it forever. It's taken us 4.5 years to get to where we are, and we've done it in partnership with all the different constituencies, and that's been a real group effort and really gratifying to see.
You mentioned data. We hear so much about AI and data analysis. When you look at something like COVID or different cancers, there's just so much data because so many people have had those diseases. With something that's relatively new and with fewer people with it, how does that affect the ability to use the data or gather the data?
Yeah, very good question. It's a multi-pronged effort. So there are AI technologies that enable us to develop a probability, a predictive probability as to whether or not a patient has a gain of function or a loss of function mutation in GRIN-NDD. And we use that, but right now the gold standard is a set of assays that have been created by a leading basic scientist out of Emory University by the name of Steve Trinellis, who looks at a number of different assays to assess the activity at the receptor. When exposed to his assays, we can get a determination as to whether or not their gain of function, loss of function are indeterminate. The number of patients that we're able to study, which I think was a part of your question, how do we find these patients? And if the population is so small, what can we do to source them? It is a bootstrapped exercise. It is a grassroots effort. We really do look at each individual family. We interact with each individual patient advocacy group to try to drive awareness. And we've been helped, to my earlier comment, by the work that's going on away from us in understanding these diseases. But if I dial the clock back 10 years and I think about why we haven't seen more GRIN, given the real world data that we have presenting to clinics, it's largely because of how these conditions have been treated historically. So to my earlier comments on the patient journey, these patients would come in and they would look like a neurodevelopmental disease patient or a developmental epileptic encephalopathy patient. And they would be treated empirically because the treatments are relatively well established. Find the symptom, treat the symptom. And it mattered a little bit less to clinicians and physicians as to what the underlying genetics were because there were no available treatments to intervene on a disease-specific basis. So they took dozens of different anti-epileptics that have either been approved and are now generic or are approved and currently branded. And they would trial these patients on those medicines and achieve really poor outcomes, but they would still work their way through a number of different empirical treatments. Same can be said around the behavioral difficulties these patients suffer from, or the sleep-wake difficulties. There are empirical interventions, but nothing that goes after the underlying disease biology. But that was the mainstay of treatment. Just identify the clinical symptom and treat the clinical symptom. And what that didn't do was drive a sense of urgency around how to understand underlying disease biology, in part because even if there was curiosity in that regard, it was very hard to get the studies that would prove it out. And so people leaned on empirical treatment, which has been true in neuroscience forever. Look at what presents in the clinic, understand the phenotype, and treat the phenotype. We want to understand the genotype and treat the genotype, which again is a part of our world in neuroscience that's gathered a lot of momentum because of the genetically defined treatments that have come to market and those that are in late stage development, irrespective of therapeutic modality. It could be a small molecule, it could be a large molecule, it could be an ASO, a gene therapy. There's been a lot of momentum developed in neuroscience where a lot of monogenetic disease tends to live. So to the question on how we find these patients, the answer is a multi-pronged answer. It's a grassroots effort, and we've been benefiting from the tailwinds that exists around whole exome sequencing. And I think the physicians, particularly those who've recently trained, have been taught to think about genotype alongside phenotype, not just phenotype. And that's benefited all of us. We'll continue to organize around this, locking arms with all the different constituencies to drive awareness, but the success of our program will have a big impact on that.
Because of those challenges, though, how did you initially approach addressing these conditions, as there weren't really that many points of reference, I guess?
No, there were not. We did it hand in glove with the patient advocacy community. And the first meeting between industry, scientists, and the patient advocates in this disease took place in 2019. And it was an effort to understand the disease through the eyes of the patient and the caregiver. What symptoms are most significant? How do they live with this condition? How do the caregivers live with the patient and provide for them and their care? And incorporated that feedback into our clinical trial design so that we were driven by the patient experience, the lived experience. And with regard to the symptoms, these are not atypical symptoms. So patients presenting with seizure, behavioral dysregulation, and the variety of different symptoms described before are pretty familiar. Some are specific to this disease, but they are not difficult to assess. So there had been pathways driven by other non-disease specific therapies that were looking to modify some of these symptoms, symptomatically, of course. So we had some breadcrumbs to follow as we organized around clinical trial design. What we did that was different was we said, historically, companies would look at a single cohort of patients and identify seizure frequency decrease in those patients when exposed to an experimental drug. And as a secondary outcome measure, they would look at the other parts of the disease to see if they got better. And that's a meritorious pursuit. It's important to understand the broader disease, but very, very hard to do when you're looking at just a seizure cohort. Because if you're decreasing seizure frequency by 40, 50, 60, 70%, and you're seeing improvement beyond seizure, really hard to disentangle whether that improvement is driven by the drug on the disease or whether it's driven by the drug on seizures and as a secondary phenomenon on other parts of the disease. So we approached our phase 1b/2a study, the Honeycomb study, in a novel way where we said, let's take two cohorts. Let's study two cohorts, one of whom has active seizures, in our case, 4 or more a month, 1 a week. And let's see if we can decrease seizure frequency while also looking at behavioral and non-seizure outcome measures. But let's take another cohort of patients whose seizures are well controlled. And let's see what the drug can do on those symptoms without having to disentangle or disaggregate post hoc the symptoms that are not seizure symptoms. And that was different and new, hadn't really been done before in this space of neurodevelopmental disease and DEEs. And we were fortunate in that we saw in the seizure cohort 86% seizure frequency decrease on top of standard of care. So these patients in that cohort came in with an average of 25.5 seizures a month, despite being on an average of 3 anti-seizure medicines. So they came in on the regimen they were on before the trial, they were left on that regimen, and the baseline number of seizures were counted. And then we titrated radiprodil and treated them. in a maintenance phase with radiprodil, and then looked to see the difference between seizure frequency on radiprodil plus standard of care versus seizure frequency on just standard of care. And that's where we saw the 86% seizure frequency decrease, which was pretty remarkable. We also saw a dramatic increase in seizure-free days, which means a lot to families and caregivers as they live their day-to-day lives. And the non-seizure cohort gave us some really important information as well. So in that cohort, patients were on an average of 1.5 anti-seizure medicines, but they had 0 seizures in the observational period, which is what we wanted. We wanted a well-controlled patient population vis-a-vis seizure. So those patients also were titrated on radiprodil and maintained over an 8-week period. And what we saw was improvement in clinician-rated CGI outcomes, caregiver-rated CGI outcomes, clinical global impression of change. And then we saw improvement over time. The longer patients were on medicine, the more they improved on a rating scale called the ABCC irritability subscale, which is a scale that's rarely used prior to our use of it in DEEs and NDDs, but has some precedent, some regulatory precedent. So risperidone, which is an old school antipsychotic, is approved to treat autism spectrum disorders with behavioral dysregulation. So as we were looking at designing the study in that cohort, we said, well, there is regulatory precedent for using this outcome measure to assess behavioral dysregulation in an ASD. That should give us some pretty valuable information. Let's use it in our honeycomb study. And what we saw was improvement over time that got deeper and more significant the longer patients stayed on drug. And that the combination of the improvement in seizure and the improvement in non-seizure outcome measures in a well-controlled cohort is really what got regulators excited about our program, because we gave them an opportunity to believe that targeting underlying disease biology, going after the receptor that is hyperactive, driven by this mutation, can have impact across the disease. Because if you go after the cause of the disease, the receptor that drives the disease, and you're having profound impact on one symptom, you'd expect to have profound impact on other symptoms of the disease. And that's what we were able to show, albeit in an open label study with a small number of patients, but the effect was remarkable enough to drive the phase 3 design in the way that we did. And now we're teed up to prove out the theory of the case, because the phase 3 study also has these 2 cohorts, one of whom has active seizure, one of whom has well-controlled or no seizures. And we'll have an opportunity to prove that out in the phase 3 study. And if we do, we'll be in a position to gain approval for the broader disease as opposed to seizures in the disease. And that's the big distinction between what we are doing and what other companies have done before us. Because, of course, as you know, companies have been approved. Rare disease, rare epilepsy, rare developmental epileptic encephalopathy drugs have been approved. But when they're approved to treat the seizures associated with the disease, not to treat the disease itself. And our goal is the latter, not the former.
And as far as the trial's concerned, when are you anticipating starting that and maybe even getting some interim results?
Yeah, so we have begun that study, that global phase 3. We are dosing patients currently. We have sites activated in the US and Europe, the UK. And we will be activating sites in Asia Pac. And those sites will come online over the course of this year. But as we continue to enroll in the US, where we know the patients, we know where they live, we know what the genetic mutation is, we've characterized it to know that it's gain of function, we've got ongoing dialogue and relationships with the principal investigators, and we're driving hard at maximizing the pace of enrollment. The way we think about the study right now, it's a 100-patient study. So 60 patients in the seizure cohort, 40 patients in the non-seizure cohort, randomized 1-to-1. And our goal is to enroll across the globe. And our expectation is that we'll have top-line data from that study in the back half of 2027.
What are your hopes for those suffering from these conditions?
We aim to have maximum benefit across the disease. So despite not targeting symptoms specifically and targeting underlying disease biology, we have impact on the symptoms. The symptoms are obviously a product of the underlying disease biology. So our goal is to be able to show benefit across the disease, and the endpoints that we have in place right now are endpoints that should help us prove that out. What does that mean, though, clinically? What does it mean day to day for these patients and these families? It means different things for different patients, different families. In some cases, patients who are experiencing significant behavioral dysregulation. There's things like self-injurious behavior and impulsive behavior. Some patients scratch themselves, bite themselves, are difficult to control for the parents and the caregivers, which, as you can imagine, has significant impact on quality of life. And in those patients, our goal is to modify those symptoms and to create more balance in the lives of these patients and families and improve their quality of life. In other patients, the primary symptom are seizures, and there isn't as much behavioral dysregulation. There may be some, but what really impacts quality of life for the patients and the caregivers are the frequency of the seizures and their ability to leave their home for simple things like family events or a brief vacation. If we have an ability to significantly decrease those seizure frequencies and increase the number of seizure-free days, we can have dramatic impact on the lives of those patients. In some patients, it's both. They've got significant seizure burden and they've got significant behavioral dysregulation and neurodevelopmental symptoms. So, of course, in those patients, we're aiming to treat both. But because we're able to go after the underlying disease biology, we don't have to pick and choose which symptoms we're aiming to treat because we're aiming to treat all of it. But for some patients, given that the population is heterogeneous, you may find that there's more of an emphasis on behavior in a certain patient and more emphasis on seizure in another. And again, because of the mechanism of action, our goal is to serve all patients with this disease who have gain-of-function mutations. And what we've heard from families coming out of our phase 1b/2a study, of course, anecdotally from parents and from caregivers, is that the impact can be pretty profound, where the dynamics that exist in the home between siblings and the patient, between siblings and the parents, between spouses, can have significant downside consequences. And we've heard that from families. And so what we've also heard from families is that coming out of the clinical trial where they've seen remarkable improvement, it hasn't just been about the patient's life, it's been about the life of the family and how everyone gets along in the home. Anecdotes about parents living better lives, marriages being rehabilitated in some way. And when you think about these kinds of diseases in that way, it really gives you a sense of how far-reaching the implications are across the family and the caregivers. So when I get asked the question of what is it that we're trying to accomplish for these patients, my answer is it's not strictly related to the patient by product of having impact on the patient or having impact on the broader family. And that's what's different about these diseases, not just GRIN-related NDD, but the broader neurodevelopmental disease family of diseases. It does have impact on everyone. And our goal is to start with the patient, but then move to the family, because if we can have material impact on the patient, we'll have material impact on the family.
You've mentioned a couple of times about being involved with groups and families. How important is it to be at that kind of grassroots level, I guess, to be involved in that?
Well, I can't overstate how critical this relationship is. I mentioned before that in the earliest days of the company, we solicited feedback from these caregivers and family members who take care of these patients. And that feedback was instrumental in designing our phase 1b/2a study. We revisited that feedback formally in a patient caregiver survey prior to our phase 3, and that feedback significantly impacted the way in which we designed the study and the way in which we engaged with regulators. The personal experiences of these families really can't be overstated in terms of the impact that it has on us. But I'd go a level deeper and say the purposefulness of the work that we do is enhanced by these relationships. The intimacy that we have with these leaders in these patient advocacy groups and the families that are a part of these groups is remarkable. And it drives the work that we do. It's part of every conversation we have at the company, how much this work matters and how individualized the work tends to be, because we know how important this is to these patients because of our interactions with the patient advocacy groups, because of what we learned from them. It's a powerful tool. These diseases are difficult. The way in which we aim to treat them and the trial designs and the execution against it is challenging. But when you're motivated by real lived experience that we are intimately familiar with, given how close we are to these patient advocacy groups, it is a huge motivator across the organization. You don't have to be in clinical development to feel motivation and inspiration from these patient advocates and patient advocacy organizations. You can be in finance, operations, business development, you can be an executive assistant, and you will derive similar motivation, energy, compassion, and purpose from your interactions with these groups. In fact, one of our earliest employees at the company is a patient advocacy leader and had started her own patient advocacy organization in a different disease. But to give you a sense of how we prioritize patient advocacy in our company, one of the earliest employees was herself a mom of a child with a neurodevelopmental disease. And so we take it really seriously. It informs the work we do, but above and beyond that, it serves as a major source of inspiration. And when we are in the trenches, engaged in hand-to-hand combat that is clinical development, drug development, we keep that in the very front of our minds, and it energizes us.
You're already doing a lot, but are there other things that you're working on?
Yeah, so we are focused on NR2B biology. And that was the organizing principle for GRIN for obvious reasons, because the GRIN genes encode for the NMDA receptor. But we stepped back and we said, where else does NR2B biology rear its ugly head as it relates to pediatric diseases? And we quickly determined that two diseases, FCD, focal cortical dysplasia, and tuberous sclerosis complex, implicate NR2B biology gone awry. They do it in a slightly different way than GRIN-related NDD, but these patients, these kids, and adolescents and even adults present with refractory seizures. They have seizures that can't be treated effectively with standard anti-seizure medicines. And when they can't be treated effectively, and the lesions that define these diseases, you can see the lesions in the brains of these patients. When they're not in elegant cortex, like the language center of the brain or the motor strip of the brain, they can be resected in surgery. So some of these patients go to neurosurgery, they have the lesions resected with the hope that the resection of the lesion will cure the epilepsy. Unfortunately, it rarely does cure the epilepsy, but it does have potential to deliver significant clinical impact. So when these patients go to surgery, then they have the tissue resected. We can study that tissue outside the brain. And in fact, leading KOLs, PIs in the space have done just that. And they took the tissue from an FCD lesion and studied it ex vivo and saw a 2 to 4-fold increase in NR2B expression, which is not shocking because NR2B expression is known to be increased in underdeveloped brain. And these lesions in FCD patients, this dysplastic tissue, being rich in NR2B, again, is, I won't say expected, but not surprising. So that's a structural validation of NR2B being a target in FCD and TSC. But when you take that same tissue and you study it in a functional assay, an assay called the zero magnesium model, and you expose the dysplastic tissue to an NR2B NAM, our mechanism of action, you see electrophysiologic normalization. When you expose the margins of that dysplastic tissue, which are not dysplastic, you don't see electrophysiologic change when exposed to an NR2B NAM. So as we were ramping for our next indication, which is FCD and TSC, we took those structural data and the functional data, and we showed it to leading KOLs, and we said, does it make sense to use an NR2B negative allosteric modulator in this patient population? And the feedback we got was it absolutely does, and it's consistent with the approach we've taken in GRIN to go after underlying disease biology. And we are in a phase 1b/2a study right now, studying both FCD and TSC patients, and we'll be in a position to report out those data in the near term. So we've been active as a company away from just GRIN-related NDD and focused on broader diseases that incorporate or implicate NR2B biology, but they're all rare pediatric diseases. So the comments I made before about the dynamic that surrounds these conditions, the patient dynamic, the family dynamic, very similar things apply as we cut across the different indications we're studying. And we're really focused as an organization around this notion of disease-specific therapies. And we think the broader neuroscience space has gotten to a point where patients deserve a disease-specific therapy. We think it can deliver more robust effect size And our goal is to prove that out.
And you mentioned an event in Edinburgh. Is that next month? What's that?
To my earlier point on how we integrate and collaborate with the patient advocacy groups, there are a number of these groups that exist in certain geographies in different countries around the world. And we spend time with all of them. So as often as we can, we are there in person. We've recently come back from a conference in France. We spent a lot of time around the US attending conferences, as you might imagine. And there's an upcoming conference, the GRIN UK conference in the UK in Edinburgh in mid-May. And we will be at that conference talking about our program, interacting with the patients and the families and the principal investigators as well who attend these family conferences. And they're really important because they provide information and an opportunity to interact with all of these constituencies where they can ask questions and better understand the work that we do, get better oriented to what a clinical trial is, why we do it, how much we care about safety and protecting the patient, while at the same time doing our work to deliver a novel therapy. It's also a wonderful social experience because you get to see the real life experiences of these patients and families. And in many cases, the patients come with parents to these conferences when the conditions that they experience are consistent with traveling. They're there and we can see them interact. I'll share with you an anecdote. An 11-year-old brother of a GRIN patient approached me at a conference this past July in Florida. And it was as if I was talking to a 20-year-old or a 25-year-old young person. The level of sophistication that this young man had at the age of 11 years old, the sensitivities that he had developed being the brother of a patient with GRIN, the perspective that he developed at that age, the sense of purpose in helping to care for his sibling, and the relationship that he had with his mom, who was there by his side. I think it speaks to one of the elements of the broader experience for these patients and families that often goes unrecognized or under-recognized, perhaps, which is this learning experience that these families undertake. While there are clearly moments of pain and moments of difficulty, there are also moments where people grow as human beings. And when we go to these family conferences and we see folks like this young man who demonstrates a level of maturity and mindfulness that, again, I'd expect to see in a young adult, if they're lucky, was a really remarkable moment. And we derive a lot of gratification from attending these conferences and really look forward to them. They're energizing for us. It gives us even more sense of purpose. So we're excited to be in Edinburgh. I hope the weather holds up and we'll spend time with families there.
Another really informative discussion there. Actually, two very informative discussions this week. So that's it for this week's podcast. If you do want to get in touch with guest opportunities or if you want to advertise, please do contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.